Hello, everyone. My name is Eduardo Williams, and I participated in the Heart, Lung, and Blood Summer Research Program at Case Western Reserve University. I conducted cancer research at Dr. Ruth Carey's lab in the Cleveland Clinic and worked on a project that sought to determine the mechanism of action of cyclin-dependent kinase 7 inhibitors in triple-negative breast cancer. To provide a little bit of background, triple-negative breast cancer, or TNBC for short, is one of the four molecular subtypes of breast cancer. As you can see in figure 1A, there are luminal A, luminal B, HER2-enriched, and triple-negative breast cancers. Three of these molecular subtypes express hormone receptors that are currently therapeutically targeted and have good prognostic outcomes. However, TNBC lacks the expression of all of these receptors, making it the breast cancer subtype associated with the worst prognosis. The Kaplan-Meier curve in figure 1B shows the overall survival of TNBC patients in a Brazilian cohort over a 5-year period. And you can see that TNBC patients had a 60% survival rate compared to 80% of patients with the other breast cancers. Though there are different therapies available to treat TNBC, there is currently no targeted therapy that increases overall survival. Thus, it is extremely important to discover and characterize therapies that improve the clinical landscape of TNBC. In recent years, it has been discovered that cyclin-dependent kinase 7, or CDK7, may be an effective therapeutic target for a myriad of cancers. CDK7 is a master regulatory protein required for transcription and cell cycle progression, and it is currently targeted in clinical trials. However, the mechanism of action of CDK7 inhibitors is not completely understood. Preliminary data from our lab has shown that CDK7 inhibitors decrease cell number in TNBC, as shown here in the dose response curve in figure 2A. Additionally, we performed cell cycle analysis on cells treated with the CDK7 inhibitor, which induced cell cycle arrest at the G2M phase of the cell cycle, as shown here in figure 2B. We know that there was an increased accumulation of cells in the G2M phase of the cell cycle, as shown here, compared to the vehicle treated cells. We use propydium iodide flow cytometry to separate the amount of cells undergoing each cell cycle phase in order to perform this. And the quantification for this data is showed in figure 2C. These results suggest that the CDK7 inhibitor treated cells did not undergo a successful mitotic division, and this may be due to errors in DNA packaging that may lead to genomic instability. Based on this data, I hypothesized that CDK7 inhibitors drive cell cycle arrest and induce chromosomal instability in TNBC cells. In order to study the effects of CDK7 inhibition, we needed cell models to visualize their effect on DNA. Plasmids expressing fluorescent proteins can be used to observe the phenotypes associated with CDK7 inhibition. And I stably transfected 3 TNBC cell lines and 1 non-breast cancer control cell line with 3 plasmids, each designed to observe the cell cycle stages for DNA in cells under live cell imaging technology. And these plasmids are shown here in figure 3. 2 of these plasmids express green or red histone H2B, a protein that's required for DNA packaging. And as you can see in figure 4, I successfully transfected these TNBC and control cells with the 2 plasmids. For the purposes of this presentation, I show only 1 TNBC cell line, MDAMB 231s, and 1 non-breast cancer control cell line, which are the MCF10As. So as you can see here, on the first left-hand column, we have the cells that were transfected with the H2B GFP plasmid, which glow green. On the middle column here, we have the H2B M-cherry plasmid transfected cells that glow in red. And we additionally transfected the cells with a Fuji plasmid. And this plasmid is very interesting because it glows in different colors based on the stage of the cell cycle that the cells are in. So on the right column here, we have cells that glow yellow, and these cells represent cells in the G1S phase of the cell cycle. The ones that are in green represent cells that are in the S, G2, and M phase of the cell cycle. And lastly, The cells that are in red represent cells that are in the M to G1 phase of the cell cycle. And these cells will not necessarily glow all the time, which is something important to keep in mind. Because the Fuji plasmid is designed so that it can show the cells progressing through the cell cycle, there are times when the cells will not necessarily be in doing cell cycle progression. So this is just something to keep in mind, that we know that there are cells that may actually be transfected even though that they don't glow. From here, we tested the effects of CDK7 inhibition, and we did that by treating these cell lines with CDK7 inhibitor THC1 and visualizing the effects of the treatment via live cell imaging technology. So we did that here in figure 5A. And so here we have control cells that were treated with vehicle. And so these cells mostly had successful mitotic divisions. However, THC1 treated cells had these mitotic bridges that were formed, that are shown here. Mitotic bridges form when you have two dividing cells, but their nuclei remain connected. And so mitotic bridges are abnormal. And we found that treatment with CDK7 inhibitors lead to a higher proportion of mitotic bridges compared to our control. Figure 5B shows the quantification of these mitotic bridges, which we expressed as a percent. And so we took the total mitotic bridge events per total mitotic events that were seen in the cells. We saw that the CDK7 inhibitor caused mitotic bridges in 30% of mitoses in treated cells compared to 2.4% in control-treated cells. When these mitotic bridges remain, they may increase the susceptibility of cells to genomic instability. And this means that CDK7 inhibitors may be working to induce genomic instability in TNBC cells. Though there is more data needed to investigate this mechanism, we can conclude for now that we were successful in transfecting the TNBC and non-breast cancer control cell lines with the H2B-GFP, H2B-M cherry, and Fuji plasmids. We saw that CDK7 inhibitors cause more mitotic bridges compared to baseline levels in TNBC, which are associated with genomic instability. These cell lines will continue to be used to study the cellular mechanisms of CDK7 and the phenotypes associated with CDK7 inhibition. Again, it is really important to understand the mechanism of action of these inhibitors because they are currently being used in clinical trials to treat patients with TNBC and other cancers. In the future, I plan to perform live confocal cell microscopy in Fuji transfected cells to validate the data from the flow cytometry analysis in figure 2B using these visual models. Furthermore, I plan to perform experiments investigating whether CDK7 inhibitors induce DNA damage in order to promote genomic instability. Lastly, I will perform experiments that suppress CDK7 expression to validate CDK7 as the selected target of CDK7 inhibitors. I would like to thank Dr. Ruth Carey and Brian Webb for their mentorship throughout the summer. And I would also like to thank Dr. Monica Montano, Joseph Williams, and Milana Bay for all of their support throughout the Heart, Lung, and Blood Research Program and for funding me through the National Heart, Lung, and Blood Research Training Grant. Thank you, and I'm looking forward to any questions you may have.